In this video, we shall look into the process of meiosis. Meiosis is a process of cell division which occurs in germ cells. Germ cells are the cells present in the ovary and the testes. The process of meiosis can be divided into two stages, meiosis 1 and meiosis 2. This is a diploid germ cell. Diploid means it consists of two sets of chromosomes. In meiosis 1, this cell divides to form two new cells. In this stage, homologous chromosomes separate. What are homologous chromosomes? As you know, we have two sets of chromosomes, one from the father, one from the mother. So we have a pair of similar chromosomes. These similar chromosomes are known as homologous chromosomes. In meiosis 2, these cells further divide to form four new haploid cells. Haploid means it consists of one set of chromosome. Diploid is denoted as 2n, whereas haploid is denoted as a single n. This was a short representation of meiosis. Now we shall look into further details. As we know, meiosis is divided into two stages, meiosis 1 and meiosis 2. Each of this stage can be further divided into karyokinesis and cytokinesis. Karyokinesis means nuclear division, whereas cytokinesis means cytoplasmic division. Karyokinesis 1 involves four steps. They are prophase 1, metaphase 1, anaphase 1, and telophase 1. Similarly, karyokinesis 2 involves four phases: prophase 2, metaphase 2, anaphase 2, and telophase 2. Prophase 1 is the longest phase in meiosis. It is further subdivided into 5 steps. First one is leptotene, then is zygotene, third one is pachytene, fourth is diplotene, and the fifth stage is dikinesis. We shall now look into prophase 1. The first step is leptotene. In this stage, the chromosomes condense, they become short and thick. The next stage is zygotene. In this stage, the homologous chromosomes pair together and form a bivalent. This process is called synapsis. Pachytene is the important step in prophase 1. Here, crossing over takes place. It is also known as genetic recombination. In this step, transfer of genetic material takes place. There is exchange of genetic material between the homologous chromosomes. In the fourth step, that is diplotene, the synapsis breaks and the chromosomes start separating. In the last step, that is dikinesis, the homologous chromosomes separate completely. This is a graphical representation of meiosis 1. The first step is prophase 1, which we had a look into. In this step, the chromosomes condense, become short and thick. The nuclear membrane starts disappearing and crossing over takes place. The next step is metaphase. Here, the homologous chromosomes come and align at the center of the cell. They align along the equatorial plane of the cell. The third step is anaphase. In this step, the homologous chromosomes move away from each other and go towards the pole of the cell. In telophase 1, the nuclear membrane starts reappearing and there are 2 nuclei formed. Finally, in cytokinesis, the cytoplasm divides and 2 haploid cells are formed. In meiosis 2, this meiosis 2 is very similar to mitosis. Here in prophase 2, the chromosomes condense again and the nuclear membrane disappears. In metaphase 2, the chromosomes align themselves along the equatorial plane of the cell. In anaphase 2, the sister chromatids get separated and move towards the poles of the cell. And in telophase 2, the nuclear membrane starts reappearing and 2 new nuclei are formed in each cell. Finally, in cytokinesis, 4 haploid daughter cells are formed.